Hello, very good evening to the student who have joined. Can you kindly let me know in the chat box if my voice is visible, audible, and my screen is visible to you? Also, if you can see my video, let me know if everything is all right. My voice, the video, and also the PPT. Okay, great. Thank you so much. We will wait till it is 6 and more students join, then we will start today's session. So today it is the week 10 live session where we will discuss week 11 course content. We will wait till it is the time of the class and then we will start the session after more students join. Meanwhile, if you have any problem while listening to my voice, please let me know in the chat box. So we will wait for 5 more minutes, then we will start today's session. Today it is the week 10 live session for the NPTEL course of Organic Chemistry in Biology and Drug Development. Please let me know if everything is alright with my voice and the presentation. Okay, so I think I will start with the introductions. Okay, so very good evening to the students who have joined. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for the NPTEL course of Organic Chemistry in Biology and Drug Development. Today, it is our week 10 live session. So, we will wait till more students join and then we will start today's session. So today it is a week 10 live session where we will discuss the course content of week 11. We are going ahead with every week, okay? We are a week ahead in every live session. So since it is week 10 live session, we have to discuss the course content which involved in the week 11. So right now the week 11 of this particular course of NPTEL Organic Chemistry in Biology and Drug Development is online. So I think I will wait for some more time and then I will start the session today, the live session today. Okay, so I think I will start with the introductions again. Okay, so very good evening to the students who have joined. Please let me know in the chat box if my voice is audible clearly, and also if you can see my video and the presentation. If you cannot hear my voice properly or you cannot see the presentation, please let me know in the chat box. For the smooth streaming of the class, I have muted all of you, and also but I have opened the chat box. Okay, so if you have to let me know anything you want to tell me, you can communicate through the chat box. My name is Mousumi Deb, and I'm the PMRF fellow from the Department of Chemistry, Chemical Biology, IIT ISM Dhanbad. Today it is the week 10 live session for the NPTEL course of Organic Chemistry in Biology and Drug Development. So, like every week, I will start with few points before starting the live session. So, there will be a problem solving sessions every week. Okay, so every week there will be a problem solving session, and till your course ends. Okay, these sessions will be live on Google Meet platform, and this will be on every Tuesday from 6 p.m. to 8 p.m. Okay, so if any network disruption happens during the class, I will request all of you to wait for a few minutes. Okay, do not leave the class. If I will join as soon as possible if the network disruption happens from my side, and if the network disruption happen at your end, also you can rejoin at any time. Okay, and still if the problem network issue actually if it persists, do not worry, because the lecture video and also the PPT I will share with you after the class. Okay. During the session, the students can post their question, doubts, and queries, everything through the chat box. Okay, chat box will be the medium of communication throughout our class. So, if you have anything to tell me about any doubt, your query regarding this today's class or any class or any week or any course content, you can let me know in the chat box. But if any doubt persists after the class, the students can drop an email to me. I always share my email ID at the end of the session. So if you have any doubt, any query after the class also, you can just let me know through the email. Okay, I will answer your queries through email. And this presentation and class recording, as I have mentioned before, it will be shared with the students soon. So I hope my voice is clear. If you have any problem hearing to my voice, please let me know. So the chat box is here open, so you can let me know anytime if you have any problem listening to my voice or anything. Okay, and there may be some internet problem because the weather is not very much better here. Okay, so if any problem happens, I will try to rejoin as soon as possible, but for all the students, please do not leave the class. So today it is the week 10 live session. So every week we are going ahead with our sessions. So today it is the week 10 live session, but I will solve the week 11 course content in today's class. So what is in your week 11 course content? It is the polyketide biosynthesis. Then we will discuss biosynthesis of macrolide polyketides and introduction to virus. And then we will discuss antiviral drugs. So these all are present in your week 11 course content. Okay, and today we will solve nearly 20 questions from your week 11 course content, and these questions will not only help you to understand the week 11 course contents better, these topics better, also it will be helpful for your weekly assignments. So if you can solve this question in today's class, also if you understand how we are solving this type of questions, it will be beneficial for you, because you have to submit your assignments based on this week 11 course content. Okay, so I think we can start with the questions. Meanwhile, when more students will join, I will add them with due course of time. Okay, so I think I will wait for 1 more minute and then I will start the class. So I will show you the first question. Let me show you the first question, and then we can wait. So the first question of today's session is problem number 1. It says that dash and dash are the building blocks used in the biosynthesis of 6-deoxyerythritolide B. Fill in the blanks. So here you have to fill in the blanks which are asking for the two building blocks for the biosynthesis of 6-deoxyerythritolide B. And your options are as follows. Number A, propionyl S-CoA and methyl malonyl S-CoA. Your option number B is malonyl S-CoA and methyl malonyl S-CoA. option number C is acetyl-SCoA and malonyl-SCoA. And option number D is acetyl-SCoA and propionyl-SCoA. So you have to identify which of the following options A, B, C or D is correct for the problem number 1. Means which are the correct building blocks used in the biosynthesis of 6-deoxyerythronolide B. So read all the options very carefully, read the question very carefully, take your time and let me know which of the following you think is the correct answer for this problem number 1 in the chat box. There are 4 options A, B, C and D, and let me know in the chat box which of the following options you think is the correct one. I will show you the answer eventually, then we will discuss it. But before that, I want all of you to try to answer this question. Let me check if we have any answer for first question. Okay. Okay, no worries. Take few more minutes and then I will discuss this. Okay, let me show you the answer for the first question. So the answer will be number A, propionyl-SCoA and methyl-malonyl-SCoA. So let us see what is the 6-erythronolide B and what are the building blocks. So these are the building blocks for the mm, for your biosynthesis of erythronolide 6-deoxyerythronolide B. So this is the 6-deoxyerythronolide B. Okay, this macrocycle is the 6-deoxyerythronolide B, and this is the starting point for the erythromycin biosynthesis. As we all know, erythromycin is a broad spectrum antibiotic, and we had discussed that it falls in the macrolide class of antibiotic, which is the first generation of macrolide, and we discussed it in the very last class, right? So for the biosynthesis of this erythromycin, the building blocks is deoxyerythronolide B. It is the starting point. And what is the building blocks for this starting point? These are propionyl-SCoA and methyl-malonyl-SCoA. So this is the propionyl-SCoA. Okay, and this is the methyl-malonyl-SCoA. And both of these come together with this enzyme deoxyerythronolide B synthesis 1, 2 and 3. So these three enzymes, they participate, and for this, as you can see from the structure also, there is a kind of cyclization for this both molecules, where these both molecules participate, and they form the 6-deoxyerythrolyte B, which is the starting point for our erythromycin A. Okay, so this was your problem number 1. So remember, the propyonyl S-CoA and methylmalonyl S-CoA are the building blocks for the biosynthesis of 6-deoxyerythrolyte B. Okay. So now I will move toward the next problem. So problem number 2, it says that which of the following statements is true for secondary metabolites? Your options are: number A, they are same in all plants. Number B, they are directly involved in growth, development and reproduction. Number C, they are directly involved in ecological functions. And number D, they are produced in large quantities. So which of the following statements is true for the secondary metabolite? Read all the options very carefully and then decide which of the following will be true for the secondary metabolite. Let me know in the chat box options A, B, C or D, which of the following you think is the correct one. Then I will show you what is the correct answer, and then we will again discuss about these secondary metabolites. Which one of the following statements is true for secondary metabolite? Your options are: number A, they are same in all plants. Okay, somebody wants to join. Okay, so I was in the question number, so the first question was this. So I was in the question number 2. So the problem number 2 says which of the following statements is true for secondary metabolites. Your options are: number A, they are same in all plants. Number B, they are directly involved in growth and development and reproduction. Number C, they are directly involved in ecological functions. And number D, produced in large quantities. So which of the following statements is true regarding secondary metabolite? All of you try to answer this question in the chat box. If you have any doubt, any query regarding any question, as I always have told you, you can let me know in the chat box, okay? It may not be from today's class. If you have any doubt regarding our previous classes also, you can let me know in the chat box. And also let me know if my voice is not clear or any network problem happens. I want all of you to try to answer these questions. There is no marks today on answering the questions, but if you answer, it will be only helpful for you, because it will help you to understand the course better, and also it will clarify your doubts. So now try to answer this question in the chat box. Option A, B, C or D, which of the following you think is the correct one for the problem number 2. So let me check the chat box. Okay. Okay. Okay, no worry. We will discuss. So my voice is clear, right? Please let me know in the chat box if my voice is audible to you, and also if you can see the PPT. Hello? Yeah. Okay, thank you. Okay, now let me discuss the problem number 2. Actually the weather is not right here today, so I'm afraid I might lose the connection. So that's why. Okay, so now let us see what is the correct option for the problem number 2. So the correct statement or the true statement is number C, they are directly involved in ecological functions. So what is primary and what is secondary metabolite we have to discuss. So first we will discuss the secondary metabolite. So secondary metabolites are the compounds which are derived by the pathways from primary metabolic routes and are not essential to sustain the life of the cells. These compounds do not have a continuous production. Very often, secondary metabolites are produced during non-growth phase of the cells. Secondary metabolites are the end product of primary metabolites such as alkaloids, phenolics, steroids, essential oils, lignin, resins, and tannins. Okay. Okay. Yeah. So what is the differences between the primary and secondary metabolite we have to focus on. So primary metabolites perform physiological functions in the body like intrinsic functions. Secondary metabolites are derivatives of primary metabolites. Okay. So primary metabolites are directly involved in the growth, development and reproduction. But the secondary metabolites, no, sorry, primary metabolites are directly involved in the growth, development and reproduction, and the secondary metabolites play a role in the ecological functions. Primary metabolites formed during the growth phase due to the energy metabolism. And secondary metabolites, they formed near the secondary phase of the growth. Primary metabolites are produced in large quantities and extraction is very easy, but the secondary ones, they are produced in small quantities and the extraction is difficult. The primary metabolites are small in the same in all plants, but the secondary metabolites are different or unique to different plant species. Primary metabolites a part of basic molecular structure of an organism, but the secondary metabolites are not a part of the basic molecular structure of an organism. For example, ethanol, lactic acid, nucleotides and vitamins are the examples of primary metabolites. Pigments, antibiotics and drugs are the examples for the secondary metabolites. Now let us see the options. The first option was they are same in all plants, which is wrong because they are unique for different plants. They are directly involved in growth, development and reproduction. It is not also correct because they are involved in ecological function. The primary metabolites are directly involved in the growth, development and reproduction. And they are produced in large quantity. It is not also correct because these are produced in very small quantities than the primary metabolite. Okay, that's why the extraction of the secondary metabolites are difficult. So this was the problem number 2. Now I will move toward the next problem. Problem number 3 says linking the building blocks of in a polyketide biosynthetic pathway is similar to a reaction. What is the reaction? Your options are number A, Claisen condensation, number B, aldol condensation, number C, Diels-Alder reaction, and number D, Michael addition. So linking the building blocks of the polyketide biosynthetic pathway is similar to a particular chemical name reaction. And your options are number A, Claisen condensation reaction, number B, aldol condensation reaction, number C, Diels-Alder reaction, and number D, Michael addition reaction. You have to identify which of the following options are correct. And let me know in the chat box which of the following options you think is correct. I want all of you to participate in the class, okay? Because it does not matter even if your answer is wrong, but participation is very important because it will help only for you for understanding the course much better. So since our course is based on the organic chemistry in biology and drug development, so this question is a very good question which relates your organic chemistry also with the biological systems and your drug development processes. I will give you some time and then I will show you what is the correct answer and we will discuss it again. Try to answer this question. Which of the following you think is the correct one? Linking the of the building blocks in a polyketide biosynthesis pathway is similar to which name reaction? Claisen condensation, aldol condensation, Diels-Alder, or Michael addition? Let me check the chat box now. Then we have to discuss it all again. Okay, so I have two answers, number D and number A. Okay, let me check which of the following is the correct one. See, that is important that you are participating. Even if your answer is wrong, it doesn't matter. So let me check. So the correct answer will be number A, Claisen condensation. Now let us discuss. First, we will discuss what is a Claisen condensation reaction. What is the mechanism of Claisen condensation reaction? So we have this kind of alpha ketoesters. Okay, so this is the alpha carbon of a ester. Okay, this alpha carbon of a ester here. So we have this kind of enolate. So first, what will happen? The alpha proton. Okay, the proton which is in the alpha carbon of this ester, it will be deprotonated by a normal base like sodium. Okay, it's sodium methoxide. So this abstraction of base will generate a anion which is present in this alpha carbon, and this anion is basically stabilized by this carbonyl carbon of this esteric moiety. Okay, so this anion which is in the alpha position is behaves as a nucleophile, and we all know this carbonyl carbon of the ester, it is electrophilic. Okay, because it is an electron deficient system. Why it is electron deficient? Because it have a delta positive here, and this oxygen is delta negative. Okay, and this carbon is delta positive because the electronegativity of oxygen is greater than the carbon. So there is a gradient. Okay, which makes the oxygen here electronegative and carbon electropositive. So this carbon behaves as an electrophile here, this carbonyl carbon. So this minus charge which on the alpha carbon behaves as a nucleophile, which attacks the electrophilic carbonyl carbon of the second molecule of ester. Okay, and we have this kind of structure. Now what will happen? This negative charge, it get back here, making a double bond, and OAT group which leaves as a leaving group. We all know this OET group, OME group, and then this kind of OR group, they behaves as a very good living group. So this group will be living, and we have a beta ketoester. What is a beta ketoester? See, this carbon after the carbonyl carbon of ester is known as alpha carbon, and the next carbon is the beta carbon, where we are having now a ketone moiety which is generated by this second molecule of this ester, right? So it is a beta ketoester. Okay, so what is the driving force? This is the irreversible reaction. Okay, so now what will happen? There is a chance that it can be again deprotonate. Okay, beta ketoester has a very acidic proton which is removed by the base, therefore acidic workup is needed. Okay, so this beta ketocarbon has a very acidic proton here which is removed by the base, so acidic workup is needed for this particular reaction. So now how it relates with the polyketide biosynthesis. So see, here we have this acetyl coenzyme A. As you can see from the structure, it is the acetyl coenzyme A. So the first step for the polyketide biosynthesis is the carboxylation. Carboxylation means this acetyl coenzyme A is getting added, the carbon dioxide group is added here. Okay, and we have this malonyl coenzyme A which have a carboxylic group here added. Okay, and now the second molecule of similar malonyl coenzyme A which will have this negative charge on this alpha carbon. As you can see from here, it will attack this carbonyl carbon, as S-CoA group is as a living group. So this is the added to a malonyl coesterase A added to the priming coesterase A. So this is the Claisen condensation reaction. So as you can see clearly, the mechanism which I highlighted here, it is happening in the same phase. Okay, with the malonyl coenzyme A. Here the ester is the malonyl coenzyme A. Two molecules of malonyl coenzymes A are added together, and they are making this kind of beta keto ester. And following the second molecule of malonyl coenzyme A will come, and this kind of chain will happen. Okay, so one by one the molecules of the esters will come. Okay, and then decarboxylation will happen, and this kind of polybeta keto ester will form, and this is the polyketide, which is known as the polyketide. Okay, so remember, so at the last position, this is the particular synthesis of a orosylenic acid, which is a particular polyketide. Okay, so you do not have to memorize all this thing. So just remember, the first step of polyketide biosynthesis is the carboxylation of acetyl coenzyme A to malonyl coenzyme A. Then what we'll have, we will have Claisen condensation reaction, which creates the pathway for the polyketide biosynthesis. Okay, so remember, the Claisen condensation is the reaction. So now I will move toward the next problem. So the problem number 4, it says that the cofactor for the conversion of acetyl-CoA to malonyl-CoA is, your options are number A, NADP, number B, biotin, number C, ADP, and number D, TPP. So which of the following cofactors is involved in the conversion of acetyl coenzyme A to malonyl coenzyme A? So as you can see in the previous slide, the first step which I mentioned, it is the carboxylation, right? So acetyl coenzyme A, the structure is this. The malonyl coenzyme A, the structure is this. So we have an extra COO moiety here present in the malonyl coenzyme A from the acetyl coenzyme A. So here there is a particular cofactor which is involved in this carboxylation reaction. And as I remember, I have mentioned this thing, this coenzyme, we have discussed it in our previous lectures. It was, I think, lecture number 7 or 8. We have discussed it in very close detail. So just try to remember what I have told you. Which cofactor is involved in this carboxylation kind of reactions, or the addition of carbon dioxide or carbon dioxide fixation? Which cofactor is involved here? Let me know in the chat box which of the following you think is the correct one. I will give you 2 minutes. The cofactor for the conversion of acetyl-CoA to malonyl-CoA is, your options, number A, NADP, number B, biotin, number C, ADP, and number D, TPP. Let me check the chat box now. If you have any doubt at any point of the class, please let me know. Okay, I will discuss that as soon as possible. Okay, number B. I have one answer, number B. Let me check if it is correct or not. So, the correct answer will be, yes, very good. It is number B, biotin. So, let me show you once again. So, this is the acetyl coenzyme A. Okay, and this acetyl coenzyme A is converted to malonyl CoA by the acetyl CoA carboxylase, which is the enzyme. Okay, acetyl CoA carboxylase is the enzyme, and biotin is the cofactor which is involved in this particular reaction. And here one ATP molecule is converted to ADP plus PI, and the energy it is taken by this carbon dioxide molecule and it is added with this acetyl CoA to make this malonyl CoA. So the correct answer will be biotin. So very good. So now let us move forward. Problem number 5 says which enzyme is responsible for the termination step of the polyketide biosynthesis. Your options are number A ketoreductase, number B thioesterase, number C oxidoreductase, and number D dehydrase. So which of the following enzyme is responsible for the termination step of the polyketide biosynthesis? Is it ketoreductase? Is it thioesterase? Is it oxidoreductase or is it dehydrase? Let me know in the chat box which of the following you think is the correct answer for problem number 5. Which enzyme is responsible for the termination step of polyketide biosynthesis? Number A ketoreductase, number B thioesterase, number C oxidoreductase, and number D dehydrase. So I do not see any answer. Okay, let us discuss it. Okay. So the correct answer for problem number 5 will be sorry. So the enzyme which is responsible for the termination step of the polyketide biosynthesis is number B thioesterase. Now let us discuss it. So what does thioesterase do? So basically, the thioesterase is the examples like there are can be proteins, glutathione, acetyl ACP, acetyl CoA. So these are the thioesterases. So what are the roles? Like acetyl CoA is involved in membrane fusion, beta oxidation, gene regulation, okay, inflammation. Acetyl ACP is involved in the fatty acid biosynthesis, phospholipid synthesis. Yeah, so I will repeat again. Okay, so problem number 5 was which enzyme is responsible for the termination step of the polyketide biosynthesis? The correct answer is number B thioesterase. So there are different kind of thioesterase like acetyl CoA, okay, like which is involved in membrane fusion, beta oxidation, gene regulation, inflammation. Acetyl ACP involved in fatty acid biosynthesis, phospholipid biosynthesis, anti-tumor antibiotics and antibiotics, glutathione in lactic acid production, formaldehyde detoxification, and several proteins which are kind of thioesterase like in cellular localization. So what is the role of thioesterase? So basically it is converting a thioester to it is just actually what is basically do is normal ester, okay, and the water is converted to thiol, this coenzyme thiol. So this is breaking this bond, right, basically. So Here you can see in the fatty acid, in the last step of the termination step of the polyketide biosynthesis, the thioesterase takes part, okay, and it is responsible for the termination step. So you do not need to memorize each and every step for this particular synthesis, because this kind of polyketide biosynthesis and this kind of biosynthesis are very long, okay. So you do not memorize all these things, but actually try to remember the enzymes which are involved in the steps of the biosynthesis of this kind of polysynthesis. Few you have to memorize, but you do not need to memorize all of them. So this was the problem number 5. Now let us discuss the next problem. Problem number 6 says how many modules are involved in the formation of the macrocyclic core in erythromycin biosynthesis. Your options are number A 7, number B 8, number C 6, and number D 3. So how many modules are involved in the formation of the macrocyclic core of erythromycin biosynthesis? In the very first question today, we discussed about the biosynthesis of 6-deoxyerythrolyte B, which is the starting point for the erythromycin biosynthesis. Now this is also related to the erythromycin biosynthesis. So you have to identify which of the following is the correct. Erythromycin, if you remember, I have discussed in last class, which is a macrolide class of antibiotic, which is very important one. And erythromycin is the first generation macrolide biosyn antibiotic. So now, let me know in the chat box which of the following you think is the correct answer. How many modules are involved in the formation of the macrocyclic core? Number 7, number 8, number 6, or number 3? Let me know in the chat box. All of you try to answer this question. Even if your answer is wrong, it does not matter. Let me check the chat box. Okay, number A, I have only one answer. Okay, let me see. Okay, so the correct answer will be number C. 6 modules are involved in the formation of the macrocyclic core of erythromycin, erythromycin. Okay, so let me show you. So, the macrocyclic core of erythromycin is made by 6-module polyketide synthesis known as called DEBS, producing the lactone 6-DEB. Okay, so this is the lactone 6-DEB, which is the 6-deoxyerythrolyte B, which I have discussed. So as you can see, there are 6 modules here. So this is the starting point. Okay, and then we have this module. So we have this kind of module, module 1, 2, 3, and 4, 5, and 6. So these modules are nothing but, so there are various kind of enzymes and building blocks are involved in this, in each and every module. Okay, so as a starting point, which I have discussed in the very first question, like what are the starting points for this thing, which is propionyl CoA and methyl malonyl CoA. Okay, so this is the propionyl CoA and this is the methyl malonyl CoA, which is the two building blocks stuff to start, and then one by one the molecules. This is the methyl malonyl CoA. Okay, so first two okay, so what happens at first, there is this methyl propionyl coenzyme A. Okay, now propionyl S-CoA, two molecules are added. So one molecule is added here. Okay, and then together, this methyl malonyl A is added one by one, and this kind of chain formation happens from module 1 to module 6. And after the chain formation, what happened, the cyclization happened, and this macrocyclic core, which is a 12 carbon macrocyclic core, okay, so this formation happens. Okay, this is 2, this is 4, and all this after this formation of this macrocyclic core. So this is known as 6-deoxyerythrolyte B, which is the building block or the starting point for the erythromycin biosynthesis. Okay. Okay. So this was the first. So remember, you do not need to memorize this. Okay. So this thing is not for your memorization, but you have to remember few things like what are the starting points. The starting points are the propyonyl CoA and methylmalonyl CoA, and all the six. There are 6 modules are present. After the 6th module, what happens? This core have formation happens, which is the 6-DEB or 6-deoxyerythrolyte B, and the 6 module polyketyl synthesis is known as DEBS. Okay, so the complete synthesis is 10,283 amino acid long. Okay, so this complete synthesis is 10,283 amino acids long. 3 huge subunits are present here. These are the 3 huge subunits. Each have 2 modules, and each module have 3 to 6 domains or catalytic sites. Okay, so just remember these few points. You do not need to memorize the synthesis part. Okay, but remember these few points, like this complete synthesis have 10,283 amino acids, 3 huge subunits, each having 2 modules, means 6 module total, and this 6 module polyketide synthesis is called as DEBS, and this produce the lactone ring, which is macrocyclic ring 6-DEB, which is 6-deoxyerythrolyte B. So these are the important points which you have to remember for the erythromycin biosynthesis. Okay. So now I will move toward the next problem. Problem number 7 says the sequence of the enzymes involved in the fatty acid biosynthesis is number A, ketosynthesis, ketoreductase, dehydratase, and enolate reductase. Number B, dehydratase, ketoreductase, oxidoreductase, and ketosynthesis. Number C, enolyl reductase, dehydratase, ketosynthesis, ketoreductase. And number D, ketoreductase, ketosynthesis, enolyl reductase, and dehydratase. So what is the sequence of enzymes which are involved in the fatty acid biosynthesis? You have to identify the correct option, A, B, C, or D. So take a few minutes, and then I will show you the correct answer and we will discuss. You have to identify the correct sequence of the enzymes that are involved in the fatty acid biosynthesis. First, all of you try which of the following sequence you think is the correct one. Then I will show you and we will discuss about it. Let me check the chat box. Okay. Okay, so I have one answer, right? Number C. Okay. Let us discuss and let us see if it is correct or not. Even if it is wrong, it is all right. At least you tried to answer. So the correct answer is actually number A. The correct order or sequence of the enzymes involved in the fatty acid biosynthesis is number A, ketosynthetase, then ketoreductase, then dehydratase, then enoyl reductase. So let us check. So what are the functions of fatty acid? Fatty acids have four major physiological roles. Number one, fatty acids are the building blocks for phospholipid and glycolipids. Number two, many proteins are modified by the covalent attachment of fatty acid which target them to membrane locations. Number three, fatty acids are fuel molecules. They are stored as triglycerol, triacyl glycerol. Fatty acid mobilized from triglycerols are oxidized to meet the energy needs of a cell or organism. Number four, fatty acid derivatives serves as hormones and intracellular messengers steroids, sex hormones, and prostaglandins. So these are the functions of the fatty acid. Now let us discuss the fatty acid biosynthesis. So this is a very complicated reaction pathway. So I will not tell you to memorize everything, but from the names of the, if you understand, so remembering few things will be important. Like what is the primer? So the primer or the starting point for fatty acid biosynthesis is acetyl ACP. So this is the acetyl ACP, which is the primer, and the chain extender is the malonyl ACP. This is this. Okay. So the first step is the combination of this acetyl ACP and malonyl ACP, and which forms this 3-oxobutanoate. Okay, and this what is the enzyme participates there? It is the beta ketoacetyl ACP synthetase. Okay, or in short ketosynthetase. And what is the byproduct? This carbon dioxide and acetaldehyde. Okay, now the next step, this 3-oxobutanoate, it converted to 3-hydroxybutanoate. Okay, and the enzyme is the beta ketoacetyl ACP reductase. Then it converts NADPH to NH plus to NADP plus, and then this 3-hydroxybutanoate converts to the trans to butanoate, and the enzyme involved here is the beta hydroxyacetyl ACP dehydratase, and water is formed as a byproduct. And now this 3-hydroxybutanoate is converted to trans to butanoate. There is a double bond, and this is a transformation. And the enzyme involved here is the enoyl ACP reductase. Okay, and this converts this trans to butanoate, and NADP plus is the byproduct here. And then then this is the starting point of butanoate. Okay, and this butanoate with 6 malonyl ACP extenders, it will form with the help of thioesterase, it will form the fatty acid. It is the starting point of the fatty acid biosynthesis. This is type 1 fatty acid synthesis. So remember, these are the important enzymes. So I will suggest you to remember the names of the enzymes. That's the names of the enzymes are important, and I would suggest you to remember the names of the enzymes, like beta ketoacetyl ACP synthetase is the first one. Then beta ketoacetyl ACP reductase, which is the second one. Beta hydroxyacetyl ATP dehydratase, the third one. Enoyl ACP reductase. And also remember this thioesterase, which I have mentioned earlier also. Okay. So this is the I have mentioned, I think that the chain termination happens with the help of the enzyme thioesterase. Okay. So this was about your fatty acid biosynthesis. Now I will move toward the next problem. Problem number 8 says, which of the following statements is true regarding the virus? Number A, viruses are living organisms. Okay, so there is two times this organism, so it will be one time. This typographical error. Number B, pneumonia is a viral infection. Number C, viruses are giant compared to bacteria. And number D, viral infection is systematic. So you have to identify which of the following statement is correct regarding the virus. Virus, we are now very much used to with this virus, and after the COVID, I think we are the lots of studies are done on the virus. What is the pathways through which we are infected by the virus, and also the treatments regarding the virus. So virus is a very important topic nowadays, not only for the this course, also any kind of medicinal or biological course. So this question stems for the basic differences between the virus and bacteria. Okay. So identify which of the following statement is true. Number A, viruses are living organisms. Number B, pneumonia is a viral infection. Number C, viruses are giant compared to bacteria. Number D, viral infection is systematic. All of you try to answer this question. Option number A, B, C or D, which of the following you think is true? After you try to answer, then I will show you what is the correct answer of this question. Let me check the chat box now. Okay, so I have quite a few answers. Number A, number D, number C and number A. Okay, let us see which of the following will be correct. So, the statement which is true regarding virus is number D, viral infection is systematic. It is the correct. Okay, let me first just correct this thing, otherwise I will just okay. So, viral infection is systematic is the correct answer. Now let us discuss. What are the virus? Virus, the definition says that it is a pathogen or disease causing agent not considered living because it cannot reproduce of its own. So this is a very important point. Okay, the viruses are not living enzyme. They are not considered as a living particle. Why? Because they cannot reproduce of its own. You have to remember this thing very importantly. Okay, this is a very basic thing about the virus and most of the people got confused here. Then this is a very good difference between a very striking difference between a virus and bacteria. That the virus is not considered as a living organism because it cannot reproduce of its own, while a bacteria is considered as a living one. Okay, so what are the common key differences between these two pathogen, virus and bacteria, which are very much we actually hear about these things very commonly. So viruses are not living organisms, but bacteria, they are living organisms. Viruses only grow and reproduce inside the host cells they infect. When found outside the living cells, viruses are dormant. Their life therefore requires the hijacking the biochemical activity of the living cell. Bacteria are living organisms that consist of single cell that can generate energy, makes its own food, move and reproduce, typically by binary fission. This allows bacteria to live in many places like soil, water, plants and the human body and serve many purposes. Viruses are submicroscopic. Bacteria are giant compared to viruses. The viral infection is systematic. Viruses infect a host cell, then multiply by thousands, leaving the host cell and infecting other cells of the body. Bacterial infection is usually confined to a part of a body described as a localized infection. Infection may be caused by the bacteria or by the toxins, the endotoxin that are produced. Systematic diseases caused by viral infection include influenza, measles, polio, AIDS, and COVID-19. Bacterial diseases include pneumonia, tuberculosis, tetanus, and food poisoning. So these are the striking differences between these two very common pathogens of viruses and bacteria. So remember these things. Viruses are not living organism. They can grow, reproduce only inside the host cell they infect. Outside the host cell they cannot live of its own. Viruses are submicroscopic and their infection is systematic. Okay, they hijack basically the normal human cells or normal host cells, systems, all the systems, and all the reaction happens in the cell and infecting other cells. And the examples of the viral and bacterial infection you have to remember. So this was the differences between the viruses and bacteria. Okay. So now I will move toward the next problem. So problem number 9 says which of the following statements are false? So which of the following statement is false regarding the characteristic of good protein target for antiviral drugs? Your options are number A, it should be important to the life cycle of the virus. Option B, it should bear a little resemblance to human proteins. Option C, it should be important in the late stages of in virus life cycle. And number D, it should be common to different types of virus. So which of the following statements is false regarding the characteristic of good protein target for antiviral drugs? Read all the options carefully and then let me know. Number A, it should be important to the life cycle of the virus. Number B, it should bear little resemblance to human proteins. Number C, it should be important in the late stages of the virus life cycle. Number D, it should be common to different types of viruses. Identify the false statement regarding characteristic of a good protein target for antiviral drugs. Identify the false statement regarding the characteristic of good protein target for antiviral drugs. See, the chat box is always open. If you cannot understand a point or anything, please let me know, okay? Let me check the chat box. Okay, so I have one answer, number B. Okay, let me discuss. So, the correct answer will be number C. It should be important in the late stages of virus life cycle. This statement regarding the characteristic of good protein target for antiviral drugs is false. So, let me show you. Antiviral agents. So there are two type of antiviral agent. First, targeting the viral function, and the second, targeting cellular function of the that virus needs. So targeting viral function, it can be divided to few examples like nucleic acid polymerase. So these enzymes can be targeted while designing the antiviral agents. Okay, so nucleic acid polymerase, protease, integrase, neuraminidase. And nucleic acid polymerase can be of three types: DNA dependent DNA polymerase, RNA dependent RNA polymerase, DNA dependent RNA polymerase. So these all are the enzymes which are very important for viral function, and we can actually target these enzymes to development for the development of antiviral agent, and or otherwise we can target the cellular function that viral needs. So the key characteristic of the antiviral drugs are: number one, able to enter the cells infected with virus, interfere with the viral nucleic acid synthesis and or regulation. Some drugs interfere with the ability of the virus to bind the cells. Some drugs stimulate the body's immune system. Best responses to the antiviral drugs are in patients with competent immune system. A healthy immune system works synergistically with the drug to eliminate or suppress the viral activity. Okay. So what are the, so let's just check the all the options. So it should be important to life cycle of virus. It is correct. It should bear a little resemblance to human protein. It is also correct, because if the target, the protein target for the antiviral drug is similar to the human protein, so what will happen? The drug will not understand on which protein it should work. Should it work on the viral protein or the human protein? So it should bear little resemblance with the human protein. It should be important in the late stage of viral life cycle. No, it should not. It is, there is no hard and fast rule regarding this, and it should be common for to different types of viruses is also correct. So this number C option is false. So moving forward, I will go to the next problem. So problem number 10, it says that which of the following enzymes is responsible for processing HIV proteins during the production of new viruses? Your options are: number A, integrase; number B, protease; number C, reverse transcriptase; and number D, DNA polymerase. So which of the following enzymes is responsible for processing HIV proteins during the production of new viruses? Number A, integrase; number B, protease; number C, reverse transcriptase; and number D, DNA polymerase. Identify the correct option. Let me know in the chat box. All of you try to answer this question. Even if your answer is wrong, does not matter, but I think all of you should try. Take some time. Now let me check the chat box. Okay, so I have few answers: number C, number B, number C. So now two number C and one number B. Okay, let me see what is the correct answer. So the correct answer will be number B, protease. So protease is the enzyme, okay, which is responsible for processing HIV proteins during the production of new viruses. So the replication cycle, infection begins when the virus spike glycoprotein attached to its complementary host cell receptor. After attachment, a protease of the host cell cleaves and activates the receptor attached spike protein. Depending on the host cell protease available, cleavage and activation allows the virus to enter the host cell by endocytosis or direct fusion with the viral envelope with the host membrane. On entry into the host cell, the virus particle is uncoated and its genome enters the cell cytoplasm. The coronavirus RNA genome has a 5' methylated cap and 3' polyadenylated tail, which allows RNA to attach the host cell's ribosome or translation. So this is a particular example for coronavirus, this part. The host ribosome translates the initial overlapping open reading frame of the virus genome and forms a long polyprotein. The polyprotein has its own proteases, which cleave the polyprotein into multiple non-structural proteins. A number of the non-structural proteins coalesce to form a multiprotein replicase transcriptase complex or RTC. The main replicase transcriptase protein is the RNA dependent RNA polymerase. It is directly involved in the replication and transcription of the RNA from an RNA strand. The replicated positive sense genomic RNA became the genome of the progeny viruses. Progeny viruses are the released from the host cell by the exocytosis through the secretory vesicles. Infected carriers are able to shed viruses into the environment. The interaction of the coronavirus spike protein with the complementary cell receptor is central, determining the tissue tropism, infectivity, and species range to the released virus. Coronavirus mainly target epithelial cells. They are transmitted from one host to another depending of the coronavirus species, and this is the particular example for the coronavirus. Okay, so you don't need to memorize, but it is, I have given it as the coronavirus. We have just passed COVID, so I think it will be beneficial for you if you remember these things. So these are transmitted one to host to another depending on the coronavirus species, but either aerosol, formite, or facial oral route. Human coronavirus infect epithelial cells of the respiratory tract, while animal coronavirus generally infect the epithelial cells of the digestive tract. SARS coronavirus, for example, infects via aerosol route human epithelial cells of lungs by binding to the ACE2 receptor, and this ACE2 receptors are involved in also in the blood pressure. We all know, right? So this, so remember the this is the particular example for the coronavirus which I have given here a little bit. But remember, the protease is the enzyme that is responsible for processing the product for HIV proteins during production of new virus, which is mentioned here. Okay, you don't need to memorize everything, but remember the name of the particular protein enzyme, which is very important, the protease enzyme. Okay, so now I will move toward the next problem. So protease inhibitors are a particular class of a drug which is a very important antiviral drug for HIV. So proteases are enzymes that cleave proteins molecule into smaller fragment. HIV protease is vital for both viral replication within the cell and release the mature viral particles from the infected cell. Various protease inhibitors are developed for HIV treatment. Some of the FDA approved drugs are sacuanavir, indinavir, atazanavir, ritonavir. So these are the examples of the protease inhibitors. We will discuss the protease inhibitors in detail in later slides. So now moving forward, problem number 11 says the HIV protease enzyme is symmetrical, whereas mammalian proteases are not. What significance might this have? Number A, inhibitors of HIV protease enzyme must be also symmetrical. Number B, it may be possible to design inhibitor that prove selective for HIV protease over mammalian protease. Number C, only symmetrical substrates are cleaved by HIV protease. Number D, there is no significance. Read the question and options very carefully. I think all of you can answer this question if you read the options or question very carefully. I will repeat again, the HIV protease enzyme is symmetrical, whereas the mammalian proteases are not. What significance might this have? Number A, inhibitors of HIV protease enzyme must be also symmetrical. Number B, it may be possible to design inhibitor that prove selective for the HIV protease over mammalian protease. Number C, only symmetrical substrate are cleaved by the HIV protease. And number D, there is no significance. Let me check the chat box. Okay, so, okay, let us discuss it. So, the correct answer will be number B. It may be possible to design inhibitors that prove selective for HIV protease over mammalian protease. See, not only HIV viruses, they have this protease enzyme. Mammals or mammalian viruses or humans also have this kind of protease enzyme. So the difference between the structures, like this is symmetrical or not symmetrical, that will help to design selective inhibitors which will inhibit only the HIV protease or not the mammalian protease or the human protease. So HIV protease, as you can see, this is the structure of HIV protease. So HIV protease enzyme is symmetrical dimer made up of two identical protein units, each consisting of 99 amino acids. The active site is at interface between the protein units and is also symmetrical with twofold rotational or C2 symmetry. The enzyme has a broad substrate specificity and can cleave the variety of peptide bonds in viral polypeptide, but critically it can cleave bonds between proline residue and aromatic residue like phenylalanine or tyrosine. Moreover, symmetric nature of the viral enzyme and its active site is not present in mammalian protease, again suggesting possible drug selectivity. So this is very important. Like the symmetric nature of viral enzyme and the active site is not present in mammalian protease. It will help to discover a or design an inhibitor which will be only specific for HIV protease, not the human protease. Okay, so this was all about this problem number 11. So now moving forward, problem number 12 says which of the following is not an anti-HIV drug? Your options are number A, nevirapine, number B, didonacin, number C, dihydroartemisinin, and number D, zidovudine. Identify which of the following is not an anti-HIV drug. Your options are number A, nevirapine, number B, didonacin, number C, dihydromisnine, and number D, zidovudine. Which of the following is not an anti-HIV drug? Identify. This is the easier one, I think. So let me check the chat box now. Number C. I have one answer only. Number C. So I was expecting all of you to answer this question because I think I have discussed similar question before also. Okay, let me check. So the correct answer will be number C, dihydroartemisinin. So these are the structures of each and every drug. This is the structure of the nevirapine. This is a anti-HIV drug. Didonacin is also an anti-HIV drug. Okay. Zidovudine is also an anti-HIV drug, and dihydromisnine, it is not an anti-HIV drug. It is a anti-malarial drug. So not only the protease inhibitors are one of the particular your anti-HIV drug. There are several classes of the antiviral drug. So we will have one or two extra classes, I think, in our this course. So there I will discuss different kind of drugs which are based on the not only the antiviral. I will discuss several kind of drugs also. So it will helpful for you again then. So remember this thing. Remember this few drugs name, like what are the HIV drugs, what are the antibacterial. So remember few drug names. Okay. So now next problem. Problem number 13 says oseltamivir is a dash agent. So what is the oseltamivir is which kind of agent? Number A, anti-influenza. Number B, anti-herpes virus. Number C, anti-retroviral. And number D, antifungal. So you have to identify which of the following is the correct option. Take 2 minutes and let me know in the chat box which of the following you think is the correct one. Okay, let me show what is the correct answer. So the correct answer will be number A, anti-influenza. So oseltamivir is an anti-influenza agent. So oseltamivir or is known as Tamiflu. So it is used for the prevention and treatment of influenza caused by influenza A or B viruses. It is on the complementary list of the World Health Organization's list of essential medicine, the most important medication needed for the basic health system. As per December 15, 2010, the WHO reported 314 samples of the prevalent 2019 pandemic H1N1 flu tested worldwide show resistant to oseltamivir. The vast majority of mutation conferring resistant are single amino acid residue substitutions like histidine 271 tyrosine in N1 and the neuraminidase enzyme. In 2011, so these are the for the just only for your information, you don't need to remember or memorize. A 2011 meta-analysis of 15 studies showed that the pool incidence rate of the oseltamivir resistant subgroup analysis detected higher rates of influenza A patients, severely H1N1 subtype. So remember, oseltamivir or Tamiflu is an anti-influenza agent, which is a very common medicine for the influenza A and B virus infection. Okay. Moving forward, problem number 14 says, which of the following statement is not true? Number A, DNA virus have lower mutation rate. Number B, reovirus or RNA virus. Number C, RNA virus occurs inside the nucleus. And number D, double-stranded DNA virus are most common. So this question is based on the DNA virus and RNA viruses. Okay, you have to identify which of the following statement is not true, which of the following statement is false. Your options are number A, DNA virus have lower mutation rate. Number B, reovirus or RNA virus. Number C, RNA virus occurs inside the nucleus. And number D, double-stranded DNA virus are most common. Let me check the chat box. Okay, so I have few answers. Number A is the most frequently given answer. Okay, let us see what is the correct answer and let us discuss. So, the correct answer means the statement which is not true is number C. RNA virus occurs inside the nucleus. It is not correct. So, let us see the DNA and RNA virus. So, DNA virus are the virus that processes DNA as the genetic material. The most common type is the double-stranded DNA virus. The genome size is larger. The replication, it occurs inside the nucleus. Mutation rate, lower mutation rate. Okay. Accuracy of replication, slow and accurate replication. Types, double-stranded DNA virus and single-stranded DNA virus. Example, adenovirus, herpes virus, pox virus. Okay, these are the examples for the DNA virus. Now let us check the RNA virus. RNA viruses are the virus that process RNA as their genetic material. Most common type is single-stranded RNA virus. The genome size is smaller. These occurs inside the cytoplasm. The replication occurs inside the cytoplasm. The mutation rate is higher. The accuracy of replication, they show an error-prone replication. Okay. Double-stranded RNA virus, positive sense single-stranded RNA virus, and negative sense single-stranded RNA virus are the types for RNA virus. Reovirus, P coronavirus, so these are the examples for the RNA virus. Okay, so I think I have just correct it a little bit. Coronavirus mutation. It will be mutation. Okay. Okay. So as the answer was given like DNA virus is lower mutation rate, it is not correct. Okay, so the correct option will be number C. So what are the differences or the examples of the viruses? So I will suggest you to remember few names of the viruses which are RNA and DNA virus. The question can be given like which is kind of like this adenovirus. So adenovirus is which kind of virus? Is it DNA virus or RNA virus? Okay, so this kind of question can be given. So I will suggest for all of you to remember few examples of this kind of viruses, which are the DNA virus and which are the RNA virus. Few examples, remembering few examples, I think will be sufficient. Okay, so this was the 14th problem. So now problem number 15, it says on which receptor the T cell surface of receptor of the T cell surface the HIV binds. Your options are number is CD4, number B CD6, number C CD7, and number D CD2. On which receptor of the T cell surface the HIV binds. On which receptor of T cell surface HIV binds? Is it CD4? Is it CD6? Is it CD7 or is it CD2? Okay, let me check which of if you have given any answer. Okay, number A. So number A is the given answer for this question number 15, right? Okay, let us discuss. So the correct answer is very good. It is number A, CD4. This is basically how the infection happens, or everything is shown in this chart. So to start with is the binding. So this is the HIV virus. Okay, so this is the HIV virus. As you can see, there are various enzymes present, like there is integrase, there is a RNA present here, reverse transcriptase enzyme, capsid, and the protease. This is the capsid. Okay, and this is the protease enzymes. So these are the HIV virus, and these are the spikes, okay, through which it binds on the cell surface. So on the surface of the T cell, HIV binds to the CD4 receptor and one of the two co-receptors, CXCR4 or CCR5. This here, okay. So this is the first step, is the binding step. Now the second step is the fusion step. So what happens in this step, the virus fuses with the host cell membrane and releases its genetic material or RNA into the cell. Okay, here as you can see, it is fusing with this particular cell, host cell, and it is releasing its genetic material. So number 3, reverse transcription. The single-stranded HIV RNA is converted into double-stranded HIV DNA by the reverse transcriptase enzyme. Now number 4 is the integration. After the HIV DNA enters the cell's nucleus, the enzyme integrase cuts the cell's DNA, inserts the HIV DNA to it. Okay, so this is the fourth step, is the integration, where the integrase enzyme cuts this host cell DNA and insert the HIV DNA, which is produced by the reverse transcription, to the host DNA. Okay. Now number 5 is the transcription and translation. The enzyme RNA polymerase makes RNA copies of DNA. HIV RNA is either inserted into the new virus particles or processed and translated to the HIV proteins. And then we have number 6 step, which is the assembly. The long protein chains are cut into the individual proteins by the enzyme HIV protease. A new virus assembled with these proteins and HIV RNA. And the seventh phase is the release of new viral particle, which is released for the host cell, taking the part to cell's membrane and capable of infecting other cells. So this is the whole life cycle of the HIV inside the host cell. So I would suggest all of you to remember each and every step. The first step is the binding, then fusion, then reverse transcription, then integration, then transcription, translation, then assembly, and then release. So I would suggest to read all the steps very carefully and remember the steps. Okay, understand. I think it is very clear. Okay, if you have any problem understanding, let me know. I will repeat it again in the next class itself, or today, or at the last if I have time. But I think you will understand once you read this, all these steps very carefully. And this is the process by through which the HIV binds with the cell and infects, and new viral particles are released for infecting other cells. Okay, so this was the problem number 15. So now moving forward, problem number 16 says David Baltimore, Nobel Prize winning biologist, classified viruses in dash groups. You have to fill in the blanks. So your options are number A 4, number B 6, number C 7, and number D 8. So in how many groups David Baltimore, a Nobel Prize winning biologist, have classified the viruses? Is it 4, is it 6, is it 7, and is it 8? Take 2 minutes and try to answer this question. In how many groups David Baltimore have classified the viruses? Is it number 4 or 6 or 7 or 8? Let me check the chat box. Okay, I think I have few answers. Number 8, number D, number C. Okay, let me show and then let's discuss. So the correct answer will be number C, 7. Actually, in 7 groups, he have uh, classified the viruses. Okay, so basically, so we have typical three classes. First is the DNA virus, which have genetic material as DNA, then RNA virus, and then reverse transcripting virus. Okay, so the DNA virus are of two types. First is the group 1. The group 1 is the double-stranded DNA, and then group 2 have a single-stranded DNA virus, okay, which can convert it into double-stranded DNA virus also. Then RNA virus are classified into the three class: double-stranded RNA virus, which is group 3, single-strand RNA virus, which is group 4 and which is positive, and single-stranded RNA virus negative is the group 5. Okay, and this is the RNA virus. And reverse transcripting virus can be of two group. Number 6 is the single-stranded RNA, which is and there is double-stranded DNA, which is group 7. So these are the groups in which the David Baltimore have classified the viruses. So the total is the number 7. So now moving forward, problem number 17 says which of the following statement is true regarding HIV protease enzyme. Number A, it belongs to enzyme family of cysteine protease. Number B, it catalyzes the formation of peptide bond. Number C, active site structure contains cysteine moiety. Number D, active site structure contains aspartic acid. So which of the following statement is true regarding HIV protease enzyme? Number A, it belongs to the enzyme family of cysteine protease. Number B, it catalyzes the formation of peptide bonds. Number C, active site structure contains cysteine moiety. Number D, active site structure contains aspartic acid. Which of the following statements is true regarding HIV protease enzyme? Number A, it belongs to enzyme family of cysteine protease. Number B, it catalyzes the formation of peptide bonds. Number C, active site structure contains cysteine moiety. Number D, active site structure contains aspartic acid. Let me check the chat box. Okay, I have few answers. Number D, number A. Okay, let me check. So the correct answer will be number D. Active site structure contains aspartic acid. It is the true statement regarding HIV protease enzyme. Okay, sorry. So this is the HIV protease enzyme. Okay, and if you can see carefully, like this active site structure, it has a active. So this is the active site mechanism of particularly the HIV protease. Okay, so as you can see, there are two aspartic acid residues, and both are connected with a water molecule which acts as a nucleophile. Okay, and this is the so active site where it acts. Okay, so just you do not memorize this mechanism. Okay, memorize, you do not need to memorize, but remember, like this active site structure, it contains the aspartic acid residue. Okay, and these are the HIV protease inhibitors which are marketed for the HIV. These are the drugs, FDA approved drugs, which are the HIV protease inhibitors. Okay, so this is indinavir, then there is a nelfinavir, ritonavir, sacuanavir, and amprinavir. Okay, so these are the drugs which are the HIV protease enzyme inhibitors. Okay, so now moving forward, Okay, this question you have to read it very carefully. Problem number 18. It says that the following structure is for saquanavir. Okay, so this is the saquanavir. So this is a HIV protease inhibitor. The decahydroisoquinoline ring system in the protease inhibitor was used to replace an amino acid residue that is present in the lead compound. Okay, this is the decahydroisoquinoline ring. So we know all the what is the structure of isoquinoline. So this is similar to quinoline. So nitrogen will be here for isoquinoline, as you can see. So this is a decahydroisoquinoline ring. So this ring system in the protease inhibitor was used to replace an amino acid residue that is present in the lead compound. Which amino acid was replaced? Your options are number A, phenylalanine, number B, aspartic acid, number C, proline, number D, tryptophan. So one amino acid residue was present here, okay, and then after the drug development process, the SRA modifications, this decahydroisoquinoline ring was introduced in place of that amino acid. Now you have to identify which amino acid was replaced. Is it phenylalanine, is it aspartic acid, is it proline, or is it tryptophan? Look at this structure very carefully, and now then decide this structure is similar to which amino acid. If this ring has replaced one amino acid, the structure must be similar with this particular ring. So in the following option, phenylalanine, aspartic acid, proline and tryptophan, which of the following amino acid have a structure similar to particular this ring? If you can identify that much, you will able to answer this question very easily. Read the question carefully, see the structure carefully, and then identify. I will give you 2 minutes for this answer, for answering this question. Take some time. Read the question, see the structure very carefully, and then decide which of the following is the correct answer. Let me check the chat box. I will discuss this very thoroughly. Okay, don't worry. Okay, I have few answers. So one answer is number C. Let me check. So the correct answer will be very good. It is number C, proline. See, the question, if you read the question carefully, it says decahydroisoquinoline ring, which is this one, was used to replace an amino acid residue. So if you remember the structure of the proline, what is the structure, what is the particular amino acid proline? As we all know, this is the structure of proline, right? There is a COOH group here present. Oh, this can be this or this and this. So proline is the only amino acid which is cyclic or imine kind of. So isn't the structure of this particular ring is very similar to proline? If you look at the carefully at the structure of proline and this ring, so you will understand the why proline was first it was proline and then it was replaced and it was introduced to this ring. So what is the reason? Saquanavir is sold under the brand names like Invirase and Fortivas. Saquanavir binds to the active site of viral protease and prevents the cleavage of viral polyproteins. So this is the SAR of the saquanavir, how the drug development happens. This is only for your understanding. You don't need to memorize everything. So as you can see, each and every part is particularly color is given. So first P1 ligand. See yellow one. This yellow one is a P1 ligand. It's so unsubstituted benzoyl group is preferred at this position. Larger group like pyridyl, methyl, piperidine, moiety interact with the S3 side of the particular HIV protease enzyme. Okay. So this is green part. It is the P3 ligand. The large hydrophobic group can be accommodated at the S3 subside of the protease. In the following generation of the PIs, P3 ligand has been removed because the necessary activity or interaction outside the substrate envelope. This purple one is the P2 ligand. Asparagine residue was replaced both to reduce the peptide character and the increase binding affinity. Carbamate and uridyl linker increase the metabolic stability. Good asparagine mimics are like thiazole, methyl, moiety, RTV, cyclic urea can be given, and this is the for the particular side. This THF substituent increases the binding affinity via strong hydrogen bonding and van der Waals interaction to the S2 subside. Okay. P1 dash ligand decrease the peptide character. See, this is the particularly deca hydroisoquinoline, right? So the proline was here first. So P1 proline has been replaced with the bicyclic deca hydro isoquinoline in the saquanavir. Why? Here it is mentioned clearly. Okay. So this is important. P1 first proline was here in the first generation, and after SAR it was replaced by the bicyclic decahydroisoquinoline. Okay, in other protease inhibitor, different hydrophobic groups have been used like phenyl isobutyl, tertiary leucine. Use of the sulfonamide carbamate link the P1 group, improve the pharmacophoric capability for the better oral viability. So this proline ring was removed and this group was introduced to make it more hydrophobic so that better oral bioavailability and pharmacophoric properties can be improved. So this group, black one is the P2 dash ligand. Different hydrophobic alkyl aromatic groups are tolerated. Cis amino hydroxyindene moiety can lead to 100-fold increase in the potency. Thiazole moiety can increase the solubility, and different kind of moieties can be optimized for the optimal hydrogen bonding and potency. And this bluish part is the peptide bond transmission binder, which is OH group, crucial for mimicking the catalytic transition state, forming a free hydrogen bonds with the aspartic acid 25 and 25 dash, which are important for the active site in the protease, which I have mentioned in the last slide. Dihydroxyethylamine, hydroxyethane groups are can use to transition state mimic. So this is the structure of the particular saquanavir, and each and every part interacts with a particular area of the protease enzyme of HIV, and here it is mentioned all the parts, okay, where it interacts, what are the interaction and how it modifies. Okay, so this reaction I give this question so that you can, I didn't understand how the SAR happens like this for this kind of protease inhibitors. Okay, so this was a question. It was a bit out of your course, but I thought to include this as well so that you can understand how the SAR happens in this kind of HIV protease. Now, problem number 19 says, which of the following statement is false regarding darunavir? Option A, it is HIV protease inhibitor. Number B, it binds with aspartate 25 and 25 prime while interacting with HIV protease. Number C, it has R configuration. Number D, it is synthesized by Professor Arun Ghosh. So what is the which of the following statement is false regarding darunavir? Read the options carefully. Let me know in the chat box which of the following you think is the correct answer. Which of the following statements is false regarding darunavir? Okay, let me check the chat box. Okay, think. Okay, let us discuss. So the correct statement will be, or the correct answer means the false statement is number C. It has R configuration. So darunavir, it is also, this is the structure of darunavir. It is a HIV protease inhibitor which binds with the aspartate 25 and 25 prime while interacting the HIV protease. Okay, and it is synthesized by Professor Arun Ghosh. That is why it is called darunavir. As you can see, the name itself has his name to give him okay, respect. So it has his name included, and this has actually R configuration. It actually has S configuration, not R configuration. So this is a particular synthesis scheme for the darunavir. You don't need to memorize it. I just have given for your understanding. Okay, the synthesis scheme, you do not need to memorize it, just for your know. So you will see how step by step this kind of protease inhibitors are synthesized from this scheme. It is particularly cleared. So now last question of today's session, it says only DNA antisense molecule that has been approved as an antiviral drug is number A nevirapine, number B formivircin, number C darunavir, number D zidovudine. So you have to identify which of the following is the only DNA antisense molecule that has been approved as antiviral drug. Nevirapine, formivircin, darunavir or zidovudine. This is the last question of today's session. Now I want all of you present, try to answer this question. Even if it is wrong, but doesn't matter. But if you know, please write the answer which you think is the correct. Let me check the chat box now. Okay, so number B and number D. Okay, let me check. So the correct answer will be number B. Formivircin is the only DNA antisense molecule that has been approved as antiviral drug. So antisense oligonucleotide therapy. So this is the DNA. After the transcription, the DNA forms mRNA, and mRNA, after translation, it forms the disease-associated protein. So what does this antisense drug do? After transcription, it actually, the antisense drug acts as a oligonucleotide. Okay, and it forms, it actually forms bond with the mRNA. This oligonucleotide or antisense drug form bond with the mRNA, and thus RNA degrades the RNA and no translation happens. Since the translation does not happen, no disease associated proteins are produced. Okay, and this is the structure of the oligonucleotide of the formivircin, which acts as the DNA antisense molecule and the antiviral drug. It is the only antiviral drug which is the DNA antisense molecule. Okay, so remember this. So this was the last question of today's session. So today what we did, we discussed about the antiviral drugs, and before entering into the antiviral drug, we have discussed into the polyketide biosynthesis and also the biosynthesis of several biomolecules. Okay, so this was your week 10 live session. And what we discussed about the week 11 course topic. So I will share my email ID with you like every day. If you have any doubt or any query regarding today's class or regarding any point of your course. Okay, now our it is at the toward the end we have reached. We will have two more classes we will have. Okay, in the next class we will discuss the last week or the week 12 session. And for the last class what I will do is I will take one question each from each week, so it will be 12 question. And again, and after that I will discuss the course content which I think I have to emphasize on. So for the last class we will have a discussion kind of class. We will have few questions, but most of the things we will discuss in detail. Like antiviral drug I will discuss in detail, like enzyme kinetics and what we have learned so far. So if you have any topic you want me to summarize in the last class, please let me know beforehand. Accordingly I will prepare the slides. Okay, so this is my email ID. So any point, any topic you want me to discuss in the last class, which will be on, I think your last class will be on 17th October. So please let me know before that class, like which topic you want me to summarize. So it was a week 10 live session for your course. So for the next class we will have on next Tuesday. I will request all of you please join and try to join on time. Our time is from 6 p.m. to 8 p.m. So please try to join the class on time. So and for today I will close the session. Okay, and if you have any doubt, any query regarding today's class, please let me know in this email ID. Okay, thank you all for joining. I will close the session for today. And again, if you have any doubt, feel free to contact me. So thank you so much. I will close the session for today now.